The overall goal of the following experiment is to investigate the deformability of different cell types using a simple microfluidics-based assay. This is achieved by fabricating a polydimethylsiloxane, or PDMS, microfluidic device to probe the timescale of cell transit through a sequence of micron-scale constrictions. Pressure-driven flow is then used to drive cells through the microfluidic channels, which enables the deformation and time-dependent relaxation of individual cells to be assayed. Next, the automated image analysis program is run in order to process the videos and obtain a histogram of the transit time data. The results show that different cell types exhibit differences in cell deformability based on their transit time through a series of constrictions.
The main advantage of this technique over existing techniques, such as atomic force microscopy or micropipette aspiration, is that microfluidic devices can successfully operate with high throughput capacities. While other microfluidic devices can also be used to assay cell deformability, we design our device so that cells passage through sequential constrictions, a geometry common in physiological contexts, such as the pulmonary capillary bed.
To begin, design the microfluidic device by selecting the width of constriction array channels to be approximately 30 to 50% of the average cell diameter and the channel height to be at least 50% of the cell diameter. Include a filter at the entry ports to remove debris and disaggregate cell clusters. Before commencing the experiment, fabricate the device master using standard techniques for lithographic micromachining. Verify the height of the patterned features using a profilometer.